That's so annoying.
Okay, ladies and gentlemen, let's get started. Hello, your Unit 9 notes. Again, we are approaching the end of new stuff for this unit. All right. Yesterday, we were starting to talk a little bit about linked genes, which I said is kind of our last new topic in this unit. What does it mean to say that genes are linked? Yeah, they're physically located on the same chromosome. How can they become unlinked? Yeah, remember, crossing over happens during prophase 1 of meiosis. So in the development of egg cells or sperm cells or any kind of reproductive cells, homologous chromosomes, so the one you got from mom and the one you got from dad, find each other. They form this big bundle of chromosomes that gets all twisted up, and pieces of chromosomes swap places, right? And in that process of crossing over, genes that were originally on the same chromosome might switch places and no longer be on the exact same chromosome. So that's how you can get those recombinant offspring, even if genes are on the same chromosome and are linked. Today we're going to talk a little more about that idea. So think about that idea of crossing over. Here, this chromosome has 5 genes labeled on it, A, B, C, D, E. Turn and talk to somebody real quick. Looking at how this chromosome is laid out, where do you think crossing over is least likely to occur? Between which 2 genes? And where do you think it's most likely to occur, and why? Turn and talk to somebody next to you about that for a second. So one of the things you need to remember to answer this question is that crossing over happens, first of all, in almost every chromosome at least once, every time meiosis happens. Secondly, the exact location where crossing over happens is random. It could be anywhere along the chromosome. Looking at these 5 genes, where do you think crossing over is least likely to happen? Emma? Why?
They're so close together, they'll be hard to decipher.
Yeah, so if crossing over could happen here, 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 every one of these places is equally likely all along the chromosome, the chances of it happening at this one, which is the only line that I drew between those 2 genes, is 1 out of however many lines I just drew. Right? It's a very small chance that crossing over would happen just precisely between those 2 genes. Where is crossing over most likely to happen on this example? And which other one? Can go further. Yeah. It's actually least likely to happen most likely to happen between A and E. Right? Because there are zillions of places. Crossing over could unlink A and E here, or here, or here, or here, or anywhere in between. There's a zillion places in there the crossing over could happen and separate genes A and E. The farther apart 2 genes are on a chromosome, the more likely they are to be separated from each other because of crossing over. Does that basic idea make sense? It's kind of the premise for everything else we're doing today, right? So crossing over is at the beginning of prophase. So the first thing that happens during meiosis is that the homologous chromosomes find each other. So like this is the one you got from mom and this is the one you got from dad. And in the beginning of meiosis, they find each other. And sometimes these two parts of the chromosome kind of swap places. So like this one and this one will get cut off and this piece will get attached here and this piece will get attached over there. Does that help? Any other questions at this point? Okay, so the important thing to remember here is the closer together the genes are, the less likely that crossing over will happen right precisely at that spot. The farther apart they are, the more likely crossing over is going to unlink those genes. And the reason that's important is we can use the recombination frequency, how often crossing over happens between pairs of genes, to construct what's called a genetic map or a linkage map, which shows the order of genes along a chromosome relative to each other. It's not a perfect map, which I will show you a little more in a little bit, but it tells us the relative order. This gene must come first, and then this one along a chromosome, based on how often crossing over happens between those genes. So we figure this out by using the recombination frequency. So if they take, if they do a cross like the fruit fly cross that we were looking at yesterday, or there was a slide we looked at with pea plants, remember some of those offspring. So if you take one parent who's heterozygous for a couple traits, and you do a test cross, if these genes are linked, most of the offspring, so say the dominant genes are linked and the recessive genes are linked. So the two dominant are on one chromosome, the two recessive are on another chromosome. Most of the offspring are either going to look like one pair. So most of the offspring are going to get the two dominant alleles from this parent or the two recessive alleles, right? And they're also going to get other recessive alleles, but we don't really care about those. That's why we do a test cross. These are the parental types that are going to look like their parents. This one's going to look just like this parent. This one is going to look just like that parent. Make sense? But if crossing over unlinks these genes, you will get some that are like this, and you'll get some that are like this, that show one dominant and one recessive trait. Those are the recombinant offspring. And by actually experimentally doing these crosses with different combinations of traits and looking at what percentage of the offspring end up being recombinant, they can actually measure and say, these two genes, gene A and gene B, tend to cross over. Crossing over happens between those genes 7% of the time. Because when we do this cross, out of all the offspring, about 7% of them show one of these recombinant phenotypes. Does that kind of make sense? That's where they get the data that they use to make these maps. I'm going to leave that for a minute. Have to erase that before I put the rest of the words up. So the recombination frequency gives us an idea of how far apart these genes are located, because the more often they cross over, the farther apart those genes probably are. So scientists know that you can relate the recombination frequency to the distance apart on the chromosome. And so we can use this data to make what's called a linkage map that's based on this recombination data. This isn't a precise measurement of physical distance or an exact number of base pairs. Because it's based on recombination frequency, we make these maps based on a unit called a map unit. This was invented specifically for this type of mapping, but one map unit is equal to a 1% recombination frequency. So we say if they're 1 unit apart, there is a 1% chance that crossing over is going to happen between those two genes. If they're 5 map units apart, there's a 5% chance that crossing over will happen between those genes. That basically makes sense? Because then what scientists will do is they will do a series of experiments. If they know that several genes are located on the same chromosome, for example, genes D, E, F, and G are all located on the same chromosome. They'll do a variety of experiments like that test cross that I just kind of sketched up here. And in one of the experiments, they will just look at trait G and D. And they'll do a test cross and they'll figure out, okay, what percentage of the offspring were recombinant, showed those recombinant phenotypes. In this case, they found an 11% recombination frequency between gene G and D. Then they did another experiment and looked at G and E and found that those two showed a 7% recombination frequency. So they gathered this data through experiments. Then what we can do, and what we're going to spend a lot of class today doing, is we can use this data to come up with a linkage map. This type of mapping I think is kind of fun, and if you're a person who likes logic puzzles, think like Sudoku type thing, this will hopefully make sense to you and you will think it's kind of a fun little game, because really what we're trying to do now is we're just trying to take this data and figure out how these genes are located relative to each other along the chromosome. We'll work through this one together. You're going to be doing a bunch of practice ones later, so you'll have a chance to try this. Usually the easiest place to start is with the genes that are furthest apart. And then there's some measure of guess and check in here. So we'll just start on this, and we'll see how it goes. So I'm going to start with this one. We know G and F are 22 map units apart. I'm actually not going to draw it on a chromosome because I think it's easier just to do this along a line, and then I'll show you kind of what it means on a chromosome later. If you want to draw this along the chromosome, that's totally fine too. This is also the type of thing that I would highly recommend that you do in pencil, because sometimes there is a little bit of guess and check and you have to try something and see if it works. And you might need to erase it and start over, okay? So first, we know G and F are 22 map units apart. I'm going to go like this. I'm going to say here's G, here's F. This distance is 22. All right, that one's done. Then you start trying to fit in the other genes around there. So we know from G to E is 7 map units. So I have 2 options, right? If G to E is 7, we could either go 7 to the right of G and say E is right about here. We could also go 7 to the left and put E over here. So we kind of have to look at the other data. In this case, it's nice when they give you the distance. So if I'm trying to figure out where E is, it's really nice when you have all the data, which I think we do here. We know that from G to E is 7 and E to F is 15. What's 7 plus 15? 22. So that tells us it has to be in between. Does that make sense to everybody? So we put E right about here. So now we know this is 7 and this is 15, because now my total from here to here is still 22. So now we've accounted for this data point and this one. So the last thing we need is point E, or D, sorry. And it tells us G to D is 11, and D to E is 4. So where would you place D? 4 units to the right of E. So D would go right about here, because then this distance is 4, which means this is down to 11. This is a total of 15. And that makes G to D total of 11. Does that make sense? I also just did one other thing there that this didn't specifically ask me to. The one distance it doesn't give us is from D to F. But I just figured it out when I took those 4 off of the 15. Does that kind of make sense? So sometimes the question will ask you, what's the distance from D to F? And you can figure it out by mapping out the genes. And the answer in this case is 11. That wasn't a question here. This now is our gene map. The reason I didn't draw it on the chromosome, here are the ways that this is not perfect. Remember, linkage maps give us a relative order of where these genes are in relation to each other. It does not tell us, first of all, it doesn't tell us how many map units along the chromosome is. It doesn't tell us if they are down at this end, G, E, D, F, or if they're up here, G, E, D, F. It is also absolutely correct if you flip this thing completely 180 degrees over, because all of the data would still fit, right? If we turned our map around and had F, D, E, G, all the numbers would still work, right? And that's important to remember when we do these practice problems. If I write my answer and it's like mirror image of yours, you're not wrong. As long as what you drew fits the data and the distances are correct, this just maps them out relative to each other. It doesn't tell you whether they go from left to right or right to left, and it doesn't tell you exactly what part of the chromosome they're found on. Does that make sense? Okay, there's one more practice problem in the notes, and then I'm going to give you some more to try on your own here. So let's do this one. Here we have genes A, B, G, and H on the same chromosome. The distances are given in map units. So we need to figure this one out. And this one does also ask, what is the distance in map units from B to G? So we're trying to figure out one additional one here. So you can start kind of working through this on your notes. I'm going to start working through it on the board. We'll see if we end in the same place. So you can take a look. And remember, if yours go G-A-B-H, also fine. Does anybody have any questions about this one? This one does also ask the distance in map units from B to G. So once you get them all laid out, then we know B to A is 10 and A to G is 2. So you know the distance from B to G. If they did an experiment with those two genes, which they didn't at this point, but if they did, we would predict a recombination frequency of about 12% recombination between B and G. Questions? All right. So we're going to take some time to practice this. For those of you in class, I have paper copies so you can actually write and do your mapping right on the worksheet by the problem. So you can come up and just to save paper, I printed one question separately. So most of them are on here, but the last one's on here. So take one of each of those and you can get started. Kylie and Ashley, this worksheet is posted in Classroom. So you can just pull it up. It's not editable, so just work some of the problems in your notes. Stay on the Meet for a little while because if people get stuck, we'll go over some of these later. But if you want to mute out for a couple minutes, that's fine. But we'll come back together in a little while and kind of see how people are doing on these, okay? All right.